Hello and thank you for your time. My name is Ana Unang and I'll be presenting my final year project 1 titled Effect of Ammonium Chloride on Laccase Production in Recombinant Pichia pastoris. Laccase can also be known as benzyl oxygen reductase or urushiol oxidase, belongs to a family of blue multicopper oxidase. It has 4 copper atoms per molecule in the catalytic center. It can be found in various plant, fungi, and bacteria, and is used in various industries such as food processing, textile, and bioremediation process. The recombinant Pichia pastoris is a host microorganism to produce laccase due to its heterologous expression system. The heterologous expression system is tightly regulated by alcohol oxidase gene 1 promoter. Due to the limited production of native laccase, it is proved to be a challenge for the industrial demand. Hence, by using recombinant Pichia pastoris, the intensification process of laccase production can be performed. The optimization of recombinant laccase production is still ongoing. There are many areas remain unexplored. The information on the effect of ammonium chloride as nitrogen source for laccase production by using recombinant Pichia pastoris GS115 is still limited. Hence, the effect of ammonium chloride as nitrogen source for laccase production needs to be studied. Objectives are to study the effect of ammonium chloride as additional nitrogen source for laccase production in recombinant Pichia pastoris GS115 and to investigate the optimum concentration of ammonium chloride needed for laccase production in recombinant Pichia pastoris GS115. Laccase was first demonstrated in Japanese lacquer tree and since then been found in various ascomycetes, basidiomycetes, and white rot fungi. Laccase has high redox potential, however low stability against high temperature and pH. The structure of laccase can be divided into 3, which are type 1, type 2, and type 3 copper. The blue color of the enzyme is generated from type 1 copper due to high absorbance of the bond between copper atoms at 610 nanometer. Type 1 and type 2 copper position close to each other where they exhibit a trinuclear cluster, and this is where the process of oxygen reduced to water occur and has been considered as green catalyst. It has broad application in industries such as dye decolorization in textile, bioremediation process in treating atrazine in soil, and wastewater treatment. It is also used in food processing industry, where laccase can degrade phenolic compound in the fruit juice. This causes the fruit juice to be long-lasting in terms of color and aroma. Pichia pastoris is a metallotrophic yeast which uses methanol as its sole source of carbon. This yeast has been used for the production of single-cell protein, which is a high-protein animal feed. Over time, it is introduced as host microorganism for heterologous expression system. The enzyme alcohol oxidase in Pichia pastoris used to oxidize the methanol into formaldehyde, which is the sole source of energy for methanol consumption. The advantage of using Pichia pastoris as host microorganism is because it can be grown on medium with high cell density and efficient extracellular protein secretion, and the presence of tightly regulated promoter AOX1, making it a host of microorganism with high-level expression system. The medium formulation and optimization is important before the scale-up production of primary metabolite. In this case, the primary metabolite is the recombinant laccase. The recombinant laccase production is affected by the nutritional requirement in the medium and the growth condition of the Pichia pastoris. Example of nutritional requirement needed is the carbon source, which either glycerol or methanol, and followed by that is the nitrogen source, which is also important to grow the Pichia pastoris. Buffered minimal glycerol medium will be used as the protein expression medium, which contain the following yeast extract, peptone, potassium phosphate, yeast, nitrogen base, biotin, and glycerol. Ammonium chloride with concentration of 0.5%, 1%, and 2% will be added to different culture medium. The control medium will not be added with ammonium chloride at all. Instead, it will be autoclaved and added with potassium phosphate as buffer, yeast nitrogen base, biotin, and glycerol. The fermentation process of the Pichia pastoris will be using the BMMY medium, which is the buffer minimal methanol medium. It will be performed in batch mode using shake flask filled with 5 to 10% inoculum, and then it will be left incubated at 30 degrees Celsius until the OD reach 226. Then the harvested cells will be resuspended in the BMMY medium until the OD reach 1.0 to induce expression. The BMMY medium contains the following yeast extract, peptone, potassium sulfate, yeast nitrogen base, biotin, and methanol. The culture will be then transferred into a shake flask and left incubated at 30 degrees Celsius for 8 days. Then, pure methanol will be added to a final concentration of 0.5% every 24 hours. Sampling will be done every 4 hours to measure the cell growth. Laccase assay will be using the ABTS method where it will emit intense blue-green color. The reaction mixture composed of the ABTS substrate, sodium acetate buffer, and the supernatant. The reaction mixture will then be incubated for 5 minutes, and then absorbance at 420 nanometer will be taken using spectrophotometer. Enzyme activity of laccase will be measured and determined by the given formula. At the end of this study, it is expected that the effect of the ammonium chloride as the nitrogen source for the production of laccase by recombinant Pichia pastoris will be understood. Apart from that, the optimum concentration of the ammonium chloride used for laccase production will be determined. Last but not least, this study will give useful insights for the optimization of laccase production in order to meet the industrial demand.